The cell cycle is essential for life by allowing cells to divide. It is composed of interphase, mitosis, and cytokinesis.
Interphase is composed of the G1, S, and G2 stage. During G1, also known as GAP1, proteins and enzymes are produced to synthesize organelles and prepare for the S stage. The S stage is where the DNA is synthesized. The G2 stage, also known as GAP2, makes sure that the cell is ready to undergo mitosis.
Mitosis is the process of cells dividing into two genetically identical somatic cells. It is important because it allows organisms to grow by making new cells as well as replacing old cells. It ensures that genetic material is able to carry out its proper function through passing information onto the new daughter cells. Mitosis has 4 stages: prophase, metaphase, anaphase, and telophase.
In prophase, the nuclear envelope begins to break up and disappear and the chromosomes condense, allowing them to become visible. The chromosomes are in pairs of genetically identical sister chromatids, which are attached to each other by a centromere. The centrioles start to move to opposite poles and they are produced by spindle fibers.
In metaphase, the sister chromatids line up on the metaphase plate around the center of the cell. This alignment is caused by the opposing kinetochores attached to each single chromosome.
During anaphase, kinetochore microtubules that are bound to the kinetochore pull each sister chromosome to opposite poles of the cell. While these kinetochore microtubules shorten, the non-kinetochore microtubules lengthen, allowing for the cell to elongate.
Telophase is the final stage of mitosis. During telophase, the chromatids are still at the opposite poles of the cell. A new nuclear membrane begins to form around the chromosomes. The chromosomes start to decondense while the spindle fibers disappear.
Cytokinesis is the splitting of the cell contents. Actin that is released by the metaphase plate walks along myosin, causing the cell to pinch together. A cleavage furrow is formed. When the cleavage furrow touches the other side of the cell, it fuses together, causing 2 genetically identical diploid daughter cells to be formed, each with the same number of chromosomes as the parent.
Meiosis is the process of splitting a eukaryotic organism's chromosome number in half to create sex cells called gametes through 2 cell divisions. Meiosis is essential for sexual reproduction and allows for genetic variation to occur. During meiosis I, homologous pairs are separated, while during meiosis II, sister chromatids are separated.
During prophase I, chromosomes condense and detach from the nuclear envelope. Pairs of homologous chromosomes, each with 1 chromosome from each parent, line up closely together forming tetrads. It is at this point that the homologous recombination or crossing over occurs at the chiasmata. These new DNA combinations allow for the genetic variations. Centrioles migrate from one another while nuclear envelope begins to break down.
During metaphase I, the tetrads align at the metaphase plate. The centromeres of the homologous chromosomes are oriented towards opposite poles of the cell.
During anaphase I, the homologous pair is split, leaving the sister chromatids to move towards opposite poles with the help from the kinetochore microtubule.
At the beginning of telophase I, each half of the cell has a haploid set of chromosomes. Each chromosome still consists of 2 sister chromatids. The chromosomes decondense and the nuclear envelope reforms.
Cytokinesis, or division of the cytoplasm, occurs simultaneously with telophase I. The process of cytokinesis in meiosis I is similar to cytokinesis in mitosis, except each cell contains sister chromatids instead of a single chromosome.
At the beginning of prophase II, a spindle forms and the nuclear membrane breaks up. However, chromosomes do not replicate. Instead, the chromosomes, still attached to their sister at the centromere, move towards the metaphase II plate.
At metaphase II, the genetically different sister chromatids line up at the metaphase II plate. The kinetochores of each sister chromosomes face the opposite poles of the cell.
During anaphase II, the proteins holding the sister chromatids together break down, allowing the single chromosomes to separate and move towards opposite pole.
Telophase II, nuclei form while the chromosomes decondense.
Cytokinesis is similar to meiosis I, except each cell contains a single chromosome. The result of meiosis is 4 daughter cells, each with half the number of chromosomes as the original parent.
This is the process of dividing a cell into 2 diploid cells which have identical genetic information and the same number of chromosomes as the parent cell. On the other hand, meiosis is the process of a cell dividing into 4 genetically unique haploid cells containing half the number of chromosomes as the parent cell. The cells produced by mitosis are somatic, while cells produced by meiosis are sex cells.